Hello, I'm Dr. Aruna Saman, and in this video, we are going to discuss replication of DNA. In order to understand the process of replication, let us first recall the structural features of DNA. DNA is a double helix and it has two parallel strands coiled around each other. When uncoiled, it looks like a ladder. The two strands are complementary to each other. The two strands run in opposite direction. One strand runs in 5' to 3' direction and the other from 3' to 5' direction. Thus, they are antiparallel. Each strand is made up of nucleotides. These nucleotides of each strand are connected by phosphodiester bonds, whereas these two strands, they are connected to each other by hydrogen bonds. First, we will understand a rough outline of the process. During replication, the DNA undergoes uncoiling. The two strands of DNA separate due to breaking of hydrogen bonds, and then they move apart. Each parental strand acts as a mold. Against each parental strand, fresh nucleotides get attracted and they are connected lengthwise by the formation of phosphodiester bonds. Thus, new complementary daughter strands are formed against each parental strand. At the end, we get two DNA molecules which are exact copies or replicas of the parental DNA. Hence, the process of duplication is called replication. Each new DNA strand has half its part from parental DNA. Here we can see in green color the parental DNA, and the other half is freshly formed. So orange colored is the freshly formed nucleotide chain. So thus the process is semi-conservative. The process of duplication of DNA is called replication. In eukaryotic cells, the replication of DNA takes place only once in the cell cycle, and it occurs in the S or synthesis phase of the interphase of the cell cycle. Through replication, it forms two copies that are identical to it, and DNA replicates through semi-conservative mode of replication. These are the series of changes now we are going to see which we come across during replication of DNA. And we will also realize that the entire process becomes possible because of the role played by different enzymes during these steps. The different steps are activation of nucleotides. The enzyme responsible is phosphorylase. Then there is initiation point. The endonuclease enzyme is active there. Unwinding of DNA molecule takes place. The enzyme responsible is topoisomerase. And then there is replicating fork formation due to the activity of enzyme helicase and single strand binding protein SSBP. And then synthesis of new strand with the help of RNA primer and DNA polymerase. We come across leading and lagging strands where DNA ligase is active, and then Okazaki fragments. Once again, we find the activity of DNA ligase and DNA polymerase alpha. And finally, we get formation of double helix, once again with the help of enzyme topoisomerase. Let us understand the first step, activation of nucleotides. The nucleotides are building blocks of DNA molecule. A typical nucleotide has pentose sugar and attached to it are phosphate group and nitrogen base. During DNA replication, the fresh nucleotides from nucleoplasm are added against the parental strand to form a long polymer of DNA strand. These freshly acquired nucleotides are present in the nucleoplasm, but they are in the inactive form. The four types of inactive nucleotides present in the nucleoplasm are deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. During replication, before polymerization, these monophosphates are activated by the process called phosphorylation. It is carried out with the help of enzyme phosphorylase. During phosphorylation, two phosphates are transferred from phosphoric group to the mononucleotides along with energy, and as a result, deoxynucleotide triphosphates are produced. Thus, we get active forms of nucleotides which are deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, and deoxythymine triphosphate. So these are active nucleotides. Thus, activation of nucleotides takes place in the beginning of the process. Next, point of origin or initiation point. Two enzymes are involved which are endonuclease and helicase. The replication always begins at a specific point O or origin and terminates at a point T. The origin is always flanked by T site. The origin occurs at a specific region on DNA that has peculiar base pair sequence. In eukaryotes, there are several origin points. Here, enzyme endonuclease creates a nick or cut on one of the strands of DNA temporarily. The nick occurs in sugar phosphate backbone or phosphodiester bond, and that initiates the breaking of hydrogen bond, which is carried out by enzyme helicase. It does its work of breaking hydrogen bonds in both directions, that is, it is bidirectional. And this resultant complex is called replicon. Such many replicons are produced in tandem or at random along the length of the replicating DNA. And then unwinding of the DNA molecule takes place. Due to breaking of the weak hydrogen bonds by the enzyme helicase, the two strands of the DNA unwind and separate. The unwinding of the strand imposes strain or negative coiling effect on the remaining part of the DNA. But this effect is relieved or neutralized by superhelix relaxing enzyme called topoisomerase. The similar enzyme in prokaryotes is called gyrase. The formation of replicating fork. Now the enzyme DNA helicase and single strand binding protein are responsible or active here. The enzyme DNA helicase operates by breaking weak hydrogen bonds in the vicinity of origin. The strands of DNA then separate and they unwind. This unwinding continues as Y-shaped replicating fork, and each separated strand they act as template. But these two separated strands are prevented from recoiling or rejoining by these single strand binding proteins. These SSBP remain attached to both separated strands. Synthesis of new strands. Each separated strand acts as a mold or template, and that is used for synthesis of complementary strand. It begins with the help of a small RNA molecule called RNA primer. The RNA primer always gets associated with the 3' end of the template strand, and it attracts the complementary nucleotides from the surrounding nucleoplasm. These nucleotides, freshly acquired nucleotide molecules, bind to the complementary original DNA strand with the help of weak hydrogen bonds. So here the weak hydrogen bonds get formed, whereas the newly bound nucleotides get interconnected by phosphodiester bond. So lengthwise connection of these freshly acquired nucleotides is with the help of phosphodiester bonds, and thus polynucleotide strands are formed. The synthesis of new complementary strand is catalyzed by the enzyme DNA polymerase. This enzyme acts only in one direction, that is from 5' to 3' direction. Leading and lagging strand. The template strand with free 3' end is called leading template, and with free 5' end is called lagging template. The process of replication always starts at 3' end of the template strand, where the RNA primer we have seen gets attached. And the new strand always is formed in 5' to 3' direction. As the two strands of the parental DNA are antiparallel, one of the newly synthesized strand develops continuously at a faster rate towards the replicating fork, and this is called leading strand. The other new strand develops in a discontinuous manner at slow rate away from the replicating fork, and this is called the lagging strand. DNA polymerase enzyme is responsible for the polymerization of these new strands. Let us understand Okazaki fragments. The DNA synthesis on lagging template takes place in the form of small fragments called Okazaki fragments. Separate RNA primers are required for each Okazaki fragment. These RNA primers are removed by DNA polymerase, and then they are replaced with the help of DNA polymerase alpha enzyme with new DNA sequences. These new DNA sequences are connected finally by DNA ligase. That is why we say that DNA ligase connects the Okazaki fragments. Formation of double helix. Finally, the DNA gyrase or topoisomerase enzyme forms double helix to form the two daughter DNA molecules. The enzyme responsible is topoisomerase. Thus, the DNA replication occurs in eukaryotic cell. Now let us discuss an experiment, Meselson's and Stahl's experiment performed in 1958. The aim of this experiment was to prove that DNA replication occurs by semi-conservative mode. That means in newly formed DNA molecule, one strand is old, 50% of the DNA is from the parental strand, and the other strand is newly synthesized. The method they used is equilibrium density gradient centrifugation technique using 6 molar cesium chloride. The salt cesium chloride forms a density gradient, a solution whose density is low at the top of the tube, and it goes on increasing steadily towards the bottom of the test tube. The methodology. Step 1. Escherichia coli bacteria are cultured in the medium containing heavy nitrogen N15. After producing few generation, the equilibrium density gradient band is obtained and the position of this band is recorded. Then in step number 2, the Escherichia coli cells are then transferred to light nitrogen containing medium N14 and they are allowed to replicate. It takes 20 minutes to get the first generation of the bacteria. A few members of the first generation of bacteria from this medium are collected. At equilibrium point, density gradient band was obtained and the position of the band is recorded. 20 minutes is the generation time for E. coli cells to grow under optimum temperature conditions. In the third step, E. coli cells were once again allowed to replicate further in the same light nitrogen containing medium N14 medium for one more generation. And the second generation in this N14 medium is obtained after 40 minutes. At equilibrium point, density gradient band was obtained and the position of the band once again recorded. Observation and explanation. In step number 1, when we cultured the E. coli bacteria in heavy nitrogen containing medium, we get the DNA having both its strand containing N15. So only one density band is obtained which contains heavy nitrogen. And being heavy, it occupies a position towards the bottom of the tube. In step number 2, the first generation of DNA in the medium of N14. Each DNA formed has parental strand having heavy nitrogen. So this parental strand is from that parental DNA and it has naturally heavy nitrogen. But against that parental strand, the fresh strand is formed using the nitrogen from the N14 medium and therefore the other strand contains N14. Thus we get the hybrid DNA having half the amount of nitrogen of N15 type and remaining half N14 type. So naturally what band we obtain is also a hybrid band and it occupies a position slightly higher than the heavy DNA or N15 band. In the step number 3, in the second generation of DNA, again in the same medium N14, we get two DNA molecules, both having N14 nitrogen and two DNA that show hybrid condition. So we get two distinct bands, one for hybrid and that occupies the same position as we obtained in step number 2. And now we have additional band and that is for light DNA containing only N14 nitrogen. And it being lighter, it occupies still higher position in this test tube. Thus, we conclude that semi-conservative mode of replication is followed, meaning that each strand in the DNA molecule serves as a template for the synthesis of new complementary strand. We have come to the end of our discussion on replication of DNA. Viewers, if you have liked this video, kindly subscribe for my channel, share your comments and press the like button. Thank you.